Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Dineen, a certified genetic counselor, and your host. So we are at the Raw Science Film Festival here in New York City, and I am honored to be sitting with the Dr. Larry Corey, who, for those that don't know, is an international renowned expert in virology, immunology, vaccine development. You've spent 25 years leading the HIV Vaccine Trials Network, HVTN, which was featured in the documentary that we got to watch called Ending HIV today at the film festival. And in 2020, you were selected by one of your good friends and colleagues of what it appears from the film, Dr. Anthony Fauci, to lead the COVID Prevention Network and run clinical trials for COVID-19. So you've had quite a remarkable career, Dr. Corey. So thank you for taking a few minutes and sitting with us.
Delighted to be here.
So from watching the film, and congratulations on the award, it won today at the festival, I'm thinking about aspects that we learned from the festival. So what makes HIV so difficult to create a vaccine as opposed to others?
It's probably multifactorial. First of all, the genetic diversity of the virus is greater than any other virus by 1,000 times. We think of COVID as, having genetic diversity. HIV's genetic diversity is 1,000 times more. And, you get as many strains in COVID as you will in one day in a HIV infection. And we've learned that not only is it magnitude of an immune response, but the breadth of the immune response. And then I think what has been unique, and I think Barney Graham in the film described it the best, to sort of say, if you're able to solve HIV, you'll be able to solve essentially all the other hard to develop HIV, non-HIV vaccines. Because the immune evasion strategies that HIV has, a lot of them were not understood and were essentially defined by what HIV does. The concept of decoy antibodies, the concept of altering proteasomal processing so that you get an immunodominant epitope, which is, neither conserved nor that variable, and that it's the subdominant epitopes, both in the T cell responses as well as certainly in the B cell responses, that are really the ones to which are the derivation of the broadly neutralizing antibodies. And it's been well recognized that you do not develop neutralizing antibodies till very late in infection. And it's the people who have the most virus around who seem to develop the mutations or the pathways for their B cells to even make broadly neutralizing antibodies. So all of these are factors. And as we understand them, we are learning how to handle them and devise strategies to, countermeasure them.
Yeah, no, it's certainly very complex, which makes sense in that you've spent, close to 3 decades on researching HIV and coming up with how can we have a vaccine for this. And part of the film goes into what are the different iterations of approaches there's been to developing vaccines. You've already mentioned T cells, of being able to ramp those up, but now is it more focused on B cells at this point?
Yeah, we started out with T cells under the assumption that T cell responses were more conserved and that, you would be able to get rid of viral infected cells. And that's sort of when we learned that even though you, let's say, put in the whole GAG gene, which was 50 kb, and put in Pol and Nef, and you gave all the entire protein of those 3 genes, the epitope to which the humans made was a median of 1 epitope per person. And it was never to the conserved regions. It was always to a variable region. So, wow, it didn't work. Those vaccines didn't work. Now, 20 years later, there's this understanding of why that happened through the proteasomal processing pathways, and there are genetics to that, and then there's ways of altering the proteasomal pathways and to come up with epitopes that will be presented and might be conserved. And so some of those processes are sort of going in a new sort of resurgence of T cell vaccines. Then there was the hope that non-neutralizing antibodies would work. And there are some vaccines, influenza is one of them, in which, there are non-neutralizing antibodies. There are only partially neutralizing antibodies that can be partially effective. And we went through that for a relatively extended period of time. We had one signal from what is called the RV14 trial that was in Thailand that showed that antibodies that were non-neutralizing to the V2 loop, the important correlate of protection. That vaccine induced it. And also the epidemic in Thailand was narrow, at least on a population basis. It was had much less diversity.
In terms of the strains?
Strains were present. So when we took it and thought we'd made a better vaccine and took it to Africa, sort of was a dismal lack of success.
Because there's more strains?
There was really more strains. And actually, it turned out that we didn't know what we were picking. We didn't expect this to happen. But the antibodies to V1, V2 were of lower magnitude on a strain basis than they were in the Thai study. So we actually didn't really duplicate the Thai study because the immunogen was not as good because the diversity was so great. We're starting to understand that a little bit. But the important issue was the antibody-mediated protection trials, in which we consciously set up some studies that were non-neutralizing antibodies or neutralizing antibodies. And we weren't making neutralizing antibody vaccines, but we had these broadly neutralizing antibodies that have been isolated from HIV-infected persons that had a lot of breadth and potency, and we infused them. And the AMP trial showed that when the viruses were susceptible and you had pretty high titers of antibodies, you actually could prevent acquisition.
Yeah, so very promising in that sense.
So, it sort of sets the track saying, look, if you can do this, the vaccine will work. It's clear proof of principle and concept. You go back to COVID, monoclonal antibodies to the COVID protein just worked. You made a vaccine that was, you only have to make a vaccine that was, 1/50th of the titer of the monoclonal, and it worked really well too, when it perfectly matched the strain. So we're pretty comfortable. So then we're left with the issue of, all right, how do you do that? HIV, we don't necessarily have those neutralizing antibodies in the germline. We gotta prime them. They have to go through a series of unusual mutations. So we actually know that you have to use structural biology to sort of nurture these B cells along. And we're doing that. And the science we're doing now is the endpoint is a B cell sequence that then makes a monoclonal antibody that sort of partially binds to a structure that will lead to a broadly neutralizing antibody. So the science is wow.
So we're really close from what it sounds like to actually have a vaccine. Are there going to be multiple vaccines though for multiple strains?
Yes.
Because that's something people probably don't understand. They're like, a vaccine, and it's almost like a cure for cancer. We're not going to have one cure for cancer. It's cancer is many illnesses within one umbrella.
The AMP trial showed that we gave a single monoclonal antibody to the CD4 binding site, and it only was effective in about 30% of the strains. We know that if you give another monoclonal antibody, let's say to the apex protein, there's one in the V2 loop, one in the V3 loop, or the fusion protein, we can get 100% coverage. Oh. So we're going to have to build a vaccine and we covered this in the film, in which we make, not just the one epitope. We have to make the 2 or 3 or 4 epitopes. It's sort of like TB. It's the way HIV therapy is. The virus has, its replicative cycle of not doing gene editing, of reverse transcription, and a lot of infidelity means you are going to get, by recombination and or random mutations, there's an enormous diversity. So you're going to have to use multiple sites of vulnerability. But there are sites of vulnerability, and that's where the field is at the moment.
Yeah. And I think just you had mentioned COVID, that you were such an integral part of this. And something that I really got out of the film that was emphasized a lot is that the research that's been done in developing vaccines, like the work you've been doing with HIV for, again, 25 years, that really created such a great groundwork to then have the vaccine for COVID be so quick. I was one of the population in the United States in that kind of first wave as a healthcare provider to protect my pregnant patients that I work with. So it just amazed me that, early December, I think it was, of 2020, I was vaccinated thanks to you and your team, which is just remarkable to be able to sit with you and say that. How did that phone call go with your close colleague, Dr. Anthony Fauci, of saying,
hey, you got to switch gears and put pause on HIV and switch to this thing called COVID.
Well, we've been talking about that. He's a close colleague and friend, and so we've been tracking the epidemic, and we sort of recognized a vaccine effort needed to be put together. It was sort of like, all right, let's, take the HIV enterprise. It was a big science enterprise. So how do we take this enterprise? And it started out with the Vaccine Research Center itself. And Barney Graham, actually cloning the spike protein and using it with mRNA. The ad vectors had all been studied already in HIV. So we had both a knowledge and an infrastructure and a relationship to the companies that were making ad vectors. And we had some early relationship with the mRNA companies. The labs, the neutralizing antibody assay that we validated for the AMP trials, David Montefiore at Duke did this, and we took this pseudovirus newt, dropped in the spike protein, and we had a validated assay for neutralizing antibodies, which was what we used to determine what the dose of the mRNAs were, what the dose of the ad vectors were.
So you had so much background work to say, oh, we're pulling from this. You're not starting from scratch necessarily. Like some people might think like, oh, we developed a new type of vaccine with mRNA, but there were years and years and years of work behind that we could really capitalize on.
Yeah, it was almost like the Japanese car manufacturing issues. There's 20 years of developing RNA. There's 20 years of developing the VRC. It was 20 years in developing the HIV Vaccine Trials Network. We had all this infrastructure. We had the biostatisticians, the people who knew how to write protocols and get it through the FDA and all the clinical trial sites. We would make just phone calls throughout the country and everybody said, yes, we'll help you out. So it was easy to expand our clinical trial site infrastructure. So we could pull off 5, ended up being 6, 30,000 person trials.
Yeah, that is just remarkable. In less than a year. Yeah. It's just absolutely remarkable. And I think about with HIV of just other ways that it's in terms of like the research and development for a vaccine has been a trailblazer in how we do science and approach research and development. So early in the film, it was covered and you were interviewed probably the most in the film. And you were also interviewing Dr. Anthony Fauci, which I thought was just really great to hear your back and forth together. But within that, talking about how engaging with the community was key in terms of the research and development. I'm someone in the queer community, so I very much I did not live through it because I'm 30, but in the sense of just looking back historically at how hard this was for the gay community, why was it so important from a scientific standpoint to say, hold on, we should be including the community and people with the condition with the research because that was not done back in the 80s, 90s.
I think, especially with a respiratory infection in which you are seeing that job exposure as well as density of living, which is associated with socioeconomic status and therefore race and diversity, were factors in hospitalization. We knew we had only one trial. We had to make this trial perfect. That's why there were 30,000 people, and that's why we insisted that each trial reflect the demography of the United States. And that there needed to be 12% Black and 25% Hispanic, and that kind of balance was needed because we were making public policy. And if you don't really have the data, does it work, quote, in this population? Not so much that vaccines are genetically determined by race, but they are genetically determined by force of exposure. If it's the supermarket workers and the meatpacker workers and the people who are out there doing the jobs that are the service industry jobs, well, they want to know if they're going to be protected. And if we were going to get compliance and we were going to get people to take it, okay. I mean, as healthcare workers, we were in chronic anxiety. We were going to take it, because historically, taking care of the sickest people, they spew the highest amounts of virus. Yes, we have some protective things, but
you're putting yourself at risk for the greater good.
And so, okay, they were going to be an easy mark, but the regular population was not going to be as easy a mark. And we actually had to demonstrate that it would work.
And build that trust and relationship, right. It sounds like. Yeah, even colleagues, I don't know if they were on your team or others, going into gay bars and talking with people like right where the community does meet. Nowadays, that sounds like, oh, of course, go where people are. But back then it wasn't. And I think I just have a big appreciation for that because we've seen with projects like All of Us Project out of NIH that it's so important to be including people and going into communities and having them be a partner in research. I also think about with, and tell me if I get this right, the Tang sessions, or how do I say that? The Tang source.
Mike, Dr. Cohn's issues of the Tang, you work on a project, you have a basic finding. Is it directly related to making a vaccine, or is it have more general purposes or other purposes? I think that was sort of, a cutesy way of saying.
Right. And the background between NASA saying we want orange juice in space, how are we going to do that? And they invented Tang, apparently. So that's how that name came to be. But as you're saying that, whenever we do any kind of research, oftentimes we end up learning a new aspect of science that we didn't intend to set out to learn. Is there anything that comes to mind that you or your colleagues have come across?
We just wrote a perspective that just came out today.
Oh, lovely. We'll link to that in the show notes.
On how HIV has helped other fields of science. And, HIV is a chronic disease. And, now let's say that one of the major reasons of dying with HIV is with cardiovascular disease. So actually did a study in which you showed statins prolonged reduced cardiovascular complications, irrespective of whether a high cholesterol. Frailty, CMV is associated with frailty, and treating CMV increased the exercise tolerance and the strength of frail HIV-infected people. Now, the issue is that unique to HIV, or are we going to see that, as the elderly, it's known that the elderly have higher CMV antibody titers and T-cell responses, but it's not been totally proven that they actually shed CMV much, that an antiviral would actually help them, and that there are sort of endothelial infections that are subclinical that we're really not recognizing. So obviously, TB and HIV are merged together. So TB drugs and TB therapies have been developed as part of the HIV research agenda. Been an enormous amount of science building on science. CAR T cells were first done in HIV-infected people. Now, they didn't have the CD28 co-stimulatory factors at that time. They didn't really work for HIV, but they showed that they were safe, and they were important in getting the field going.
There's a whole area of research of CAR Ts. We'll link to the episodes that we've done about that it's just remarkable what can come out of. And I don't want to say basic research, because this is not basic, because it's focused on HIV, but just these
sort of the translational researchers that come out of studying one chronic disease that's a complicated disease. The rhetoric that CD4 T cells are now sort of known in every newspaper article. Well,
certainly.
Where does that come from? Well, it comes from HIV.
And I think that really is timely to bring up now in terms of, you mentioned during the Q&A panel after the film that so far to your knowledge, there hasn't been a big impact in terms of funding for HIV research, which is wonderful to hear at this point, October 2025. But it certainly is being impacted in other areas of basic research, and that's going to have downstream effects. Any thoughts on just how, obviously, there's different organizations, like the Bill and Melinda Gates Foundation has been big in HIV research, but just thoughts on how important this is and that we should not be cutting the kind of funding we have been with NIH, is at least my personal view.
Well, it's certainly my personal.
Okay. I was hoping it would be, and I figured it was as a fellow scientific mind.
Yeah, I think that, we are the stellar scientific organization. The National Institute of Health is the beacon of science, and it's actually the beacon of a meritocracy. I actually do not know an area of government in which there's a greater meritocracy than in the NIH awarding of scientific grants. The National Science Foundation is the same. And it's an anathema to me that you want to tear down this meritocracy, because every country who wants to start a new scientific public funding looks at the NIH as the way to do it. We have more discoveries, more drugs, more biotechnology industries than any other country by far. And it is, to sort of deprecate that the system doesn't work, it's just not true.
That's right. That's correct. And I don't remember the amount of money. Dr. Eric Green comes on the show a lot. He's a good friend of the pod. And so he was the former director of the NHGRI. And so, him and others have said, it's like we pour $1 in and we end up getting like $2 or $3 out. So it's not even that if we're taking away all the scientific advancements, even just, financially, the money we put in, we're actually making even more in the economy. So it's not even a talking point, right? Like it can't be used against us in that sense. sense. So yeah, it's certainly something that I think all of us in science are really frustrated by, and the progress that's going to be lost, because I think our country is really a leader in this space, and we're going to have a brain drain in the opposite direction that we have been. And my other kind of controversial question, I suppose, is that the United States has the most vaccine skepticism of probably any country. How do we keep fighting the good fight to really share with the public that this is not, that vaccines help save people? You mentioned on the panel that we don't want to go back to polio, and that's something that I think all people would agree with, but especially where we have the President of the United States talking about different causes of autism that is incorrect. I mean, there's a lot of misinformation out there, and I'm wondering what your thoughts are as someone that's been working on vaccines for over 25 years. How do we turn this around in terms of the public's thoughts?
I think you need to bring people into the field who are specialists in communication and who are influencers with the people we're trying to reach. And I don't think that person is a me or the famous researcher. I think that when we have wanted to reach out to other communities with podcasters, I learned and brought in a person who is a marketing specialist for Starbucks.
Yeah.
And now we started using influencers on it, Instagram influencers with communities, and who we would bring in and educate them and see if they agreed and see if they were amenable to using their influence to get to their communities. And they were, because we weren't getting to their communities. So I think you have to sort of play the same game. And I think you're an accomplished podcaster. That's
Thank
not
you,
me.
Dr. Corey.
And so you're really valuable in these kinds of stories. So you can't get them to go on our playing field. And frankly, I've sort of recognized that my expertise and my style and how I'm being viewed is not going to change their minds, no matter how thoughtful I might ask a question or go after that. That I inherently am not going to get them to point B. I can get them to be polite, but I don't think that they will change. So their values, I think we have to approach it scientifically, actually.
And see what's worked in the past, like going into the community for HIV research worked so well. Let's do that again.
But the resistant community has a belief system, and it's going to require a different kind of person to alter their belief system.
Yeah, someone that's coming from their walk of life is helpful in that sense.
Who has embraced through their thinking, who has bought on to the issue that vaccines actually do good.
Really good.
Yeah. That mRNA is not going to imprint you into your system, that it can't alter your DNA. One cell has, almost 25,000 miles of wound up DNA. That this is not going to alter your DNA.
No, the human body is a lot stronger than that, I think, when it comes to that. And I'll have one last question, because the award ceremony is starting, and I certainly don't want to keep you from that. How close do you think we are to a vaccine for HIV? Do you think within the next 5-10 years we'll accomplish that?
I think it'll be single-digit years.
Single-digit. I like that. Even less than what I said. That's amazing. Well, thank you so much, Dr. Larry Corey. It's just such a pleasure to have you on the show. And I really appreciate you taking the time during the festival.
Okay. My pleasure.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.